So this morning's first speaker is Professor Keith Fox, who is the Associate Director of the Faraday Institute. He's also a Professor of Biochemistry at Southampton University. And he's also a trustee of the group Christians in Science, and was indeed its chairman when I was working for Christians in Science a number of years ago. His research is in DNA structure and how it's recognized by small molecules. He edits the journal as Senior Executive Editor of Nucleic Acids Research. But today he is going to talk about getting to grips with bioethics.
Well, good morning, everybody. Thank you, Ruth. Bioethics, I wonder what you understand by that term. Ruth did a rough survey recently of church leaders as to what aspects of science and faith they felt the least comfortable about, and far and away the top of that list came bioethics, and I wonder what people understood by that term bioethics. So do you want to just shout out to me what issues come to your mind when you hear that word bioethics? Stem cell research, thank you.
Genetic modification.
GM. Okay, thank you, yes. Assisted dying. Eugenics. Assisted conception, in vitro fertilization, etc., yes?
Choice at birth, selection of babies.
Okay, yes, so eugenics again, if you like. Some of those overlap with medicine, and some are specifically scientific. You've been very cautious in the list you give you. Let me take you through a long list that comes from a bioethics education project. And you can see it has everything to do with the human genome, genetic technologies, human health. The environment is a big thing, I think, in bioethics as well that wasn't mentioned. Animal ethics. And just in case that's not an exhaustive enough list, I went to another website, the Nuffield Bioethics one, and you can see there's a long, long list. So I hope you're sitting comfortably for the rest of the day. Now, what we're going to do is actually just tackle one or two from this and give one or two principles that arise from it. So where we're going, and we can take up all sorts of things in discussion and conversation, is firstly just to review how we do make ethical decisions. And for many of you, that will be totally familiar territory, and I apologize for just glossing over what I'm sure you're familiar with. We'll deal with some things to do with DNA and genetic modifications. DNA is the wonderful molecule that I've had the privilege of working on for most of my academic career. I'll say a little bit about stem cells and embryos, some other things about then gene correction and gene modification, so human genome modification, and just touch on enhancements if we've got time. And if we stop, the clock says finish and Ruth says cut before we get there, then you can bring up some of those in discussions and questions. So how do we make ethical decisions? And for a lot of people, it's just we muddle along and we make it up as we go along. For some are just what I might call the yuck factor, or some the wow factor. This is just so amazing, we have to do it, it's exciting. Or the yuck says that, oh, you couldn't possibly do that, could you? And some of you who are my age and a little bit older might remember the response to the first heart transplant was a sort of gut feeling of, oh, how can you go around with somebody else's heart in you? The yuck version of bioethics. But let's think through a bit more rigorous ways. So if you like, one way is what we might call a deontological basis. That's from Deon, rules. What rules are there out there? Now, for the Christian, the Ten Commandments obviously will be your starting point for rules. But as we all know, the trouble with rules is that people find ways to get around them. What can I get away with and still be legal? What happens when rules collide? Now, that you shall not lie, and yet you need to protect your brother or sister who will be in trouble if you don't lie. Rights fit within this as well. Human rights. The American Constitution, the right to life, liberty, and the pursuit of happiness is a kind of a rule-based thing. But we all know, too, that the letter of the Lord sometimes doesn't help. And Paul talks about the letter kills, but the Spirit gives life. We're not just rule-based people. So rules are not wrong, but in themselves they're clearly not sufficient. And risk is a big factor too in bioethics. What are the possible risks that could happen because of what we do? Other long words, consequentialist bioethics. What's the outcome? What are the consequences of anything we do? Is the consequence for human flourishing? Does it benefit mankind or not? So Jesus says, is it lawful to do good or to do harm on the Sabbath, to save life or to kill? What's the consequences of what you do? Utilitarian ethics is related to that in a way, but it's, again, what's the outcome? Is the outcome good or bad? Now, I'll give you an example of a utilitarian one, which you may not be quite so familiar with immediately. Some of the environmental bioethics. The university I'm in Southampton put a wonderful poster up that said, we are proud to be caring for the environment, we look after coral reefs. And I thought, yes. And then the subtext under it was, because it's useful for generating new antibiotics. That's a utilitarian ethic. It says we care for the environment because it's useful to us, not because it has any intrinsic worth of itself. Natural law ethics says we do what is, it helps humans reach their natural potential. It says what is natural out there? But we'll come back in a moment. We'll ask the question, well, what is natural? What is normal? There's only one normal person in this room, and that's me. And you undoubtedly think there's only one normal person, and that's you. I think coming closer to home, maybe for what influences a Christian, is that of virtue-based ethics. It's the ethical principles that bring out character, that enhance a person's character, the motivations that we have for the actions that we do. What sort of a person am I as I do these things? Bringing out the fruit of the Spirit, if you like. Fulfilling love, not just ticking boxes. Love God and do as you please, says Augustine. So the motivation of love comes at the base of these things. And of course, overarching all of this are some of the ethical principles that the medics go by. So it's doing good rather than doing harm, although often those two have to balance each other out. Autonomy, that there's some element of choice within this, some element of being able to control the direction we're going in, and questions of justice and as well. So some of the things that we talk about in bioethics relate to this wonderful molecule. I say it's the one I've spent 30 and more years working on. It is probably the only biological three-dimensional molecule that almost everybody is familiar with. It's become a bit of an icon of our age. Whether you know what it does or not, you put that up and everybody says, that's DNA, isn't it? Right? And it's an icon of our age, just as a bit of a throwaway. It's been there on stamps. It's used as a marketing tool. Don't ask me why. You can buy perfume, DNA perfume. It's got nothing to do with DNA at all. And you can buy it for him or for her. And it comes in nice helical bottles. And if you look very carefully, it's a 3-stranded helix on the bottle, not a 2-stranded helix. But it's out there in the public imagination. But what is it? Now, for those of you who understand any biochemistry and molecular biology, please forgive me. But I know there are some who maybe their biology is a little rustier. We talk about genes. What is a gene? Well, it's a region of DNA that codes typically for a protein. And that region of DNA can be between 1,000 and 1,000,000 or more of these bases long, with these A, G, C, or T, and the strict ordering of those. And within the genome, our genes are arranged on chromosomes. And a chromosome contains many thousands of those genes, one after another one, with lots of spaces in between them. In fact, 98% or more of the human genome is actually spaces between the genes. We have 23 of those chromosomes containing approximately 20,000 genes between them, 3 billion of these base pairs arranged very precisely in the human genome. Every cell in your body has exactly the same DNA in it. your DNA is slightly different from that of the person sat next to you. And different genes are switched on in different tissues at different times. So the DNA content in your brain cells is exactly the same as the DNA content in your big toe, but the proteins that are being made, the genes that are being used in those cells are very different. So what does DNA do? Is it a magic molecule? Well, it's a very boring polymer, just repeats A, G, C, and T in that very special order. It is our repository of our genetic makeup. It goes through this process of DNA copied to RNA, which is used to make proteins, and these proteins are active in different parts of the cell, and they're responsible for our metabolism, the cell structure, the organization, development, all these wonderful things in molecular biology and basic biochemistry. But actually DNA itself also does nothing. A pot of DNA just sat on the bench in front of you would do nothing. In fact, it would look like a piece of scrumpled up tissue paper, the sort that's been through the washing machine when you forgot to take it out of your pocket. It's just a boring chemical in itself, a very boring but equally extremely exciting polymer. It's the genetic material, and it's universal. All organisms use DNA as a genetic repository, and it's exactly the same polymer in each one. The sequence is different, but the nature, the chemistry is the same. And that's what brings us through to some of the genetic modification. Because it's the same overall polymer in a virus as it is in a bacterium, as it is in a banana, as it is to you and me, it's possible to move that around between different organisms. And that's where genetic modification comes into, as we take that, what is actually a boring polymer, and we take it from one organism into another one, or we manipulate the sequence of that and see what it does. And the kind of issues that arise from that are people ask, are we playing at God? Is this God's molecule? I've already said, what is natural? There are questions of risk as well as we move things from one organism to another. Are there boundaries between species that we shouldn't cross? Raises questions actually is what's special about being a human if we have the same genetic material as anything else. Who owns our biological information? Is that my own personal property? Does anybody else have a right to know what my genome is? And related to that is what rights do animals have? So let's unpick a few of those words. Playing at God. I really don't like that phrase when it's suggested that scientists are playing at God, partly because I don't know of any serious scientist who plays. Right? It is great fun doing science. It gets me out of bed in the morning, but it's not playing. It really is serious work, even though it might have amazing consequences. Playing suggests that maybe there's boundaries we shouldn't cross. People say that, no, we're made in the image of God. God is the creator. And if we're made in his image, shouldn't we be creative too and imaginative? We're told to look after the creation. Read those early chapters of Genesis, to bring it meaning and to tend for it and to care for it. And that's our mandate as Christians. Are there boundaries within that mandate that we shouldn't cross? Christians throughout the centuries have been involved in healing and restoring and feeding the poor. They're part of our Christian duty. How does that relate to any of the genetic modifications that come in? What is natural? Actually, that is not natural corn on the cob, as you're used to feeding on that. The natural form of it probably is this thing on the left, which doesn't look at all appetizing. And through various breeding over millennia, not through genetic modifications, but just by selecting, you get something that today is very good to eat and very nutritious. What is natural? What we've selected over many millennia, or what would have been out there anyway? Let me throw you out a question. Which would you rather that your foodstuffs, your wheats, etc., they were prepared by genetic modification using molecular techniques that we know now, or they were prepared by irradiating primitive strains of wheat with X-rays and treating them with carcinogens and just randomly selecting the ones that work best. Which would you prefer? The former. I've got a former down here. There's no shame in saying the latter if anyone wants the latter. Yes, what you don't realize is that most of the commercially used wheats that we have today, the high-producing ones, were generated a few decades ago by virtue of the latter technique, treating seeds with carcinogens, with X-rays, growing them up and seeing what had, just randomly, had better properties than the natural ones. And they're the statistics. The slide will be on the Moodle platform afterwards. So maybe we can be just a bit overreactive when we talk about genetic modification and compare to what we already have done in terms of what's natural. Why should we do GM anyway? This is for if you like, plants and foodstuffs. Well, agriculture is very important in that. Golden rice, a form of rice that makes vitamin A, that could be used for those parts of the world where there might be vitamin A deficiency. We can make crops that are drought resistant or pesticide resistant that should reduce the levels of pesticides that can be used. GM can be used for pharmaceuticals, for generating useful drugs. Those who are diabetic will take insulin. It used to be extracted from pigs. It was pig insulin. Now that's cloned. And it's never seen a pig or a human at all. Growth hormone. Herceptin, one of the drugs used for treating breast cancer, is generated by genetic modification. Interferon, an antiviral agent. You remember the Ebola outbreak where ZMapp was one of the antibodies that was used for that. That is actually produced by GM, and it's actually a plant product. So these things are very, very useful and out there. Why are people concerned about them? Well, the risks are there. What about the containment? What if these things get out and you get super weeds? Do they affect biodiversity? When you get monocultures of acres and acres of just the same thing being grown, that's actually not necessarily confined just to GM or of crops are largely grown in great acreages of exactly the same thing. The questions of toxicity and food safety, I think mainly that's alarmist. I think all the studies that have been done showing are these GM foods safe to eat, most of the studies show that they're unequivocally safe. So I don't think that's a problem. It has been said that actually a greater threat to biodiversity isn't GM, it's garden centres. When you think of the various plants that have been brought in from all parts of the world, bringing with them their local plant viruses, bringing all sorts of species that shouldn't normally be here. Japanese knotweed, for those of you who buy houses, and the insurance that goes with how that tunnels through concrete and is prevalent in some parts of the UK. The statistics are fascinating that in this country, 43% of people think that GM food is very or fairly harmful. That's in terms of what it does to you. And 56% across the whole of the EU think it's dangerous. Surprisingly, if you do survey the same people, 4% think that natural herbal products are very unsafe. I have some training in pharmacology and in toxicology, and if you want to talk about natural herb products being safe, I'll have a word with you afterwards. And actually within the UK and Europe, the organic lobby, rightly or wrongly, has a very great influence over the whole GM debate, and I would actually say wrongly, but we can talk about that later. It's also some of the words that are chosen to describe genetic modification. Unhelpful phrases. Frankenfood. I won't shame the newspaper that describes it as that. That's not helpful, is it? Actually, some of the words we use, genetic pollution, transgenics, mutation, genetic engineering. Engineering seems cold and calculated, doesn't it? It's the sort of building bridges rather than dealing with people and organisms. And it has to be said that a lot of people don't understand the science underpinning it. Another phrase that's used, and we'll come back to it later, is this three-parent babies. No, it's 2.0000 whatever, a tiny bit. It's mitochondrial replacement. It's not three-parent babies. It's the words that we use are sometimes unhelpful. So come back again. Why do we use GM? Food security, that's an academic buzzword which means basically feeding the world. We can do it for medical reasons, for research, for curiosity sometimes, for scientific egos, for commercial reasons, and there's nothing wrong with commercial reasons. When you go to the supermarket and buy your food, they're not selling that to you because they're concerned for your welfare. They're concerned because they're a business. And the GM is also business. And sometimes GM can be just used for fun, and I'll show you some examples of that. But we do have to ask the question, who will benefit? Will it be the powerful, the rich, or will it be the needy and the poor? And I think those are questions that we need to be asking. GM foods, one of the motivators for them is to feed the world. You know the challenges, I'm sure. 1 in 6 of the world population is hungry today, and by the time we reach 2050, it said we have to increase food production by maybe as much as 100% for what we're doing today. Can the science of genetic modification help with that in terms of boosting crop production? So we ask questions of, is it safe? Is it natural? Is it fair? Is it needed for each of those GM things? Some people may wonder about crossing the species barrier. That species are things that are set up there by God, that they're kinds, and that we shouldn't cross that between them. But actually, when we talk about species, even as biologists, it's very difficult to define what a species is. Technically, a species of two organisms can't interbreed. And the boundaries are very, very blurred, and it's something really that taxonomists love, a species. And it's not necessarily a useful concept in terms of, if you like, looking at that Genesis account and talking about kinds that God has put in place. GM could be used for what might be called xenotransplantation. Organs from one organism being used to treat humans. Hearts grown in pigs that could be used for transplants for humans. There are lots of questions with that in terms of risk, in terms of pig viruses that could be transmitted to humans. Porcine endogenous retroviruses, PERVs as they're sometimes known. We can do GM on that to take those out. So that the risks are reduced. There's a part of it is the yuck factor. Would you want to have a pig heart within you? But which is better, a pig heart or a mechanical heart? Or a failing heart? What does it say for the pigs too? People say, well, we're commodifying pigs, but we've done that already. We eat pork, or at least a lot of us do eat pork. Is it any worse to be using animals for organs than it is for meat? Pigs will be turned into spare part factories and plundered for their organs. Well, we plunder pigs for all sorts of other reasons anyway. Here's some of the fun part of genetic modification. Eduardo Keck generated this fluorescent rabbit, had green fluorescent protein in it, and he did it just as an artistic form. I think we might question as to, is that right or not? The rabbit doesn't seem to be harmed by the process, but it's a commodity. You can buy glowfish, marketed specifically to have different colors within them. They were actually designed originally for testing the quality of water, so that their color might change in maybe polluted water. That's not how they're used at all. They're an aquarium novelty. The company that produces those does have ethical principles, as you look through on their website, of making sure that it's environmentally safe, that it's humane treatment of the fish, etc., etc. And I think that's totally laudable. But it's cosmetic. It's for fun. We can look at DNA and use it for screening, for health benefits, for forensics. Those of you who travel up and back from King's Cross to Cambridge on the train will have seen adverts for 23andMe. You can send off a scraping of your cheek cells and they will send you back something about your genetic profile. Has anybody done that? You have. I keep intending to, and I'm too mean. But we can use DNA for forensics, for paternity testing, for scene of crime. We can look at it for susceptibility to disease. But does that then is that accompanied by genetic counselling? If you tell somebody that you've got a disease that may mean that you could have a debilitating disease that will come later on in life and you could pass that to your children, is there genetic counselling that goes that? Who has access to data? Come back to that in a moment. Questions of prenatal screening. Human genome was sequenced, or the first draft of it, in 2000. There's various other drafts been released since. And the statements for that was this genetic information must never be used to stigmatize or discriminate against any individual or group. I question as to whether that is inevitable from the knowledge that we have. The first human genome to be sequenced cost, I don't know how much, but it was of the order of $1 billion. Now you can sequence a human genome for the region of about £1,000. And that gives us tremendous knowledge. But who should know about that? The individual? Your family members? Should your children know about your genetic profile? Because you've passed it on to them. Would they rather remain ignorant, or would they rather know? Your GP? Should they know? Your insurers? No? Your employer? Are they going to invest in you if you've got a susceptibility to a heart condition that says, no, in your early 40s you run the risk of a heart disease? Who should know? There's no easy answers to these. And I think for many of them there's no Christian answer, but we need to at least understand the questions that can be asked. We talked about screening. It could be postnatal, prenatal, pre-implantation of what's the genetic profile of a child or an embryo, and that raises questions to when human life begins. If it's postnatal, then you have some form of a treatment that goes beyond that. If it's prenatal, then some people would advocate for abortion if there is genetic abnormalities detected. And what's an abnormality? And if it's pre-implantation, then that says, which embryos do we implant? Sometimes it's very useful. There are various ethnic groupings which are particularly prone to genetic diseases, and they can have genetic counselling as to whether they should have children or not. The Ashkenazi Jews are particularly gone through that. But do we actually run up with a risk of genetic discrimination, of the knowledge that we have discriminating against some people? And we'll come back to that in a moment. Diseases come back from DNA, and some of them are simple one-based changes in that. So things like cystic fibrosis, sickle cell anemia, thalassemia are simple 1 or 2 changes in the DNA sequence. Sometimes the DNA sequence is right, but it ends up in the wrong place, so that you get wrong expression of a gene. And many cancers are like that. Sometimes the sequence is right, but it just repeats itself rather a lot of times. And Huntington's disease is an example of that. There's the sequence C, A, and G, and you get long repeats of that. It's where the replication apparatus has stuttered in doing that. So we can find things out about these diseases. But do not get sidetracked into thinking that everything in life is in your DNA. Right? And this is a slide, I'm grateful for Dennis Alexander for putting this together. The things in the newspapers that suggest that everything is in our genes. Like the love cheat gene, the gangster gene, the mean gene, genes to hormones, the liberal gene, gluttony gene, cigarettes, it's all in your genes, isn't it? No. And the wag who wrote to The Guardian proposing a geneticism gene, a gene that predisposes some people to think that behavior is determined by genes. Most of those traits have some genetic influence, some. But a lot of them are environmental, and most of them are polygenic, and it's not one gene, it's many genes collaborating together to give a trait. So we're in danger of sometimes what I might call genetic reductionism. I feel like a genes for us fallacy. We make DNA into a god. It's got that special mystical property that we shouldn't ever tinker with, and it determines who we are. No, it may limit and restrict who we are, but it doesn't determine who we are as people. We're more than just the sum of our genes, and we do not have a genetic fatalism. Let's think through a few issues, and I can see the clock ticking on, and Ruth will begin to get jumpy in about 10 minutes. IVF. Here's the process. Egg and sperm meet together in that single-cell embryo that then divides and then implants it into the womb. And a very high proportion natural never get implanted. In vitro fertilization, you do this side of it in the test tube and then implant this into the mother. And that's remarkably successful and has enabled a number of people who would otherwise be childless to have children. That comes back, I think, to the question of rights. Is the right to have a child, is it a right? When these are made in vitro, you make many embryos, and you implant, well, it used to be implanting many, and now, of course, that's reduced so you don't get the multiple pregnancies. But what do you do with the ones that are not implanted? They're stored, and then what? And it's reviewed in this country after 10 years, but you can get some, I call them strange generations, because they're stored for a long period of time. It's actually you get a child and their mother, the effectively two siblings are a generation apart. Right or wrong? Pre-implantation genetic diagnosis takes that a little step further. Here we have again the in vitro fertilization. But at this 8-cell stage, where that 1 is divided, 1 to 2 to 4 to 8, you can then remove one of those cells and test it. And it doesn't affect the development of that embryo. It recovers very nicely from that. You can test that and then see, does that have any genetic problems? And of course, if it's healthy, which is what you want, you can implant it, and away you go, just exactly the same as before. If it's diseased, question then what you do with that. You discard it. And what is the nature of that little clump of cells? Is it human? Is it not? And there are various views on the status of that early embryo. So some would say it is a human person. And destroying it is the same as abortion. And the vigorous pro-life agenda would treat one of those early embryos as if it's fully human. Others will say it has a potential to be a human, but it isn't actually human. And there's a difference then between pre-implantation and post-implantation. Once implanted, then it has even greater potential. And others will say, well, it's just a clump of cells. It's just 8 cells. We can grow human cells in the lab, and it's just a certain kind of human cell. And I don't think there's any simple answer to that question as to just what is the status of that early embryo. You can look at some scriptural verses from the Psalms. For it was you that formed me in my inner parts. You knit me together in my mother's womb. That's post-implantation. Jeremiah, before I formed you in the womb, I knew you. Right? That's pre-implantation, but I'm not sure that Jeremiah was thinking in those terms quite when saying that. I think that says more about God's eternal control and overarching concern for us rather than necessarily about bioethics. When does the embryo become a person? Is it when sperm and eggs come together? There's no recognizable structure in that early embryo. As I said, 50% of those under normal fertilization don't implant anyway. And that early embryo can split into 2 anyway. You get identical twins. So it's clearly not one person in that early embryo. If you had the choice of saving a freezer full of early embryos or a person in a fire, which would you choose? Is it ethical not to use them? They cannot, in this country, be generated just for research. Research on embryos can only use the spare embryos left over from in vitro fertilization. They're not allowed to be used just for research purposes themselves. Let's take this 3-parent babies. It's actually mitochondrial DNA transfer. The mitochondria are the parts in your cells that generate the energy, the ATP. And they have their own genomes, and that sometimes mistakes can happen in those, and they give very debilitating diseases. They count for a very small fraction of the total DNA, and you inherit your mitochondria from your mother, not from the father at all. So in the newly fertilized egg, the mitochondria there can be diseased. And in this mitochondrial transfer, the nucleus is taken out of the cell with the diseased mitochondria and put into another cell from which the nucleus has been removed. And so here's a bit of a cartoon. The slide will be on the Moodle platform for you, so that effectively the major part of the DNA comes from the nucleus that was perfectly healthy, put into the cell body containing the mitochondria of the healthy mitochondria. So it's not really 3-parent babies. It's a tiny fraction. Of course, the question comes then that this, of course, is heritable. If that's a female, then those mitochondria will be passed on to another generation. Is this healing? We have organ transplants. This is an organelle transplant. Is that any different? I put the question to you. Stem cells were raised. These are what we call unspecialized cells that have the ability to divide and renew tissues. In all your tissues, there's a small number of stem cells there. You can extract them from the tissues. They're very specialist: kidney stem cells, brain stem cells, lung ones. They're specialists for that. They can divide for that particular tissue. They're to do with normal repair processes and growth. And there's been a lot of things you read in the media of how they might be used for therapy. Where do we get them from? Well, adult stem cells are already programmed for particular tissue types. You can, under some special circumstances, get them to revert to what might be called pluripotent stem cells. Or they're isolated from embryos themselves. So the fertilized egg here divides through 1, 2, 4, 8 cells, and further through to this one here we have then the blastocyst in here that goes on to develop the fetus. The rest of it goes for generating the placenta. This cell is totipotent. It can do everything, because it makes the placenta, it makes the fetus. Once we get to here, the name is it's pluripotent. In other words, it can't make the placenta, but it can still do lots of other things. And as the cells then divide, so they specialize. It's rather like going through an educational system. You start off, the 3 R's, and as you go through A-levels, you specialize in some things, and then you do a degree, and you specialize. And when you get further down the line, you actually can't go back to where you came from. Right? And it's exactly the same with cells as they divide. So can we use those stem cells? And it comes back again to the question about that early embryo. Francis Collins, whose name has been mentioned in some of the other talks already, says that to say that genetic engineering is unacceptable across the board because of its potential for creating some ethical dilemmas is the most unethical stance of all. It's to say we have a powerful technology which could alleviate human suffering, and we're not going to use it because we're worried about the misuses that might occur. He says, I find that completely unacceptable from every point of view. It's taken the precautionary principle too far. But we can now also take it further, begin to modify the human genome. Be nice, wouldn't it? We could take those faulty regions and cut them out and do a cut and paste and put nice ones in. Replace the diseased regions. And just so as you're familiar with the phrases, you will hear things about talons and ZFNs, and now the most potent one of the lot is CRISPR-Cas9. It sounds like a cooking oil to me, but it isn't. It's a very powerful molecular technique for being able to take pieces of DNA very precisely and replacing them. It's been done with somatic cells. That's cells that have already divided. They're not part of, they wouldn't be part of your germ cells to be passed on to the next generation, with some remarkable results. This child here this was in the newspapers, in the terminal stages of a form of leukemia, and treated with these genetically modified cells, and went to total remission. But what about modifying germ cells or embryos? Well, we've shown it can be done. Highly controversial research published back in 2015. This was not intended in any way for implanting. This is research purposes. But it's possible to actually modify the human genome. And it's been done since with correcting thalassemia, at least in cell lines. So it's powerful, but should we use it? Let me ask questions that you might want to talk about afterwards as to if we say genetic modification is free of risk, and there's always a question within that, should we modify the embryo that will otherwise die of a genetic disease? We're then giving life where otherwise there wouldn't be. Should we modify the embryo of someone that will, say, develop cystic fibrosis so they won't? There's somebody who will have a life, a restricted life, but we could modify that. We know the sequences that cause cystic fibrosis and take those damaged ones out and replace them. Should we do that? What about modifying the embryo of someone who would develop Huntington's disease, or a potential for breast cancer, the BRCA2 gene, later in life? So we're modifying it for affecting something that will happen 30, 40, maybe 50 years later. What about modifying the genome to change eye color? Right? Cosmetic. What about modifying the genome if we knew how to do it so that you could live longer? You might want to think about each one of those. Is there a slippery slope between those? Actually, at the moment, most of those gene modification technologies are not necessary because you could just go through simple pre-implantation diagnosis. You can test the embryo before implanting and then only implant the ones that weren't diseased. There are one or two cases where that's not possible. And what does it say about those who have genetic diseases? And I use the word disease loosely there. Here is Tom Shakespeare, be familiar to many of you. He talks about it's the voices of the people living with illness and impairment that need to be heard. We're not queuing up for genetic modification. He says our priority is to overcome discrimination. It raises questions about what is normal. Human diversity is part of what it is to be part of a society. And we talk about some things being a gift, a musical gift, an athletic gift. Modifying genomes then makes it more of a commodity rather than a gift and a natural talent. I'm going to give maybe 2 more slides. I've got plenty more I could say, and then I'm going to stop. But here's an example of, and it's published in the journal Nature. This girl has a form of albinism, which means she has very poor eyesight, and it's a genetic condition. And she has shown resilience. She now plays basketball. And interviewing her father and saying, now we have the ability to modify so that she wouldn't be born with that disease. Would you have done that? And he says, before she was born, yes, I would have opted for genetic modification. But now, knowing the daughter that he has, would he, with hindsight, now do it? And the answer is no. She'd be a different person. And I think tellingly, actually asking her, would you wish now that you had been genetically modified, it was possible? And the answer was no. It's the condition she had made her the person that she was, with the resilience that came through that. Another question is that there is a deaf culture. Those who are genetically deaf And it's a subculture. They have their own very precise sign languages. And there have been instances where deaf parents have asked if they could have embryo selection to select a deaf child. What would you say to that? The law said, no, you can't do it. You can only select a child that would have normal hearing. And there's a website on there you might want to look at. What is normal and what is normal, what is abnormal. We have our own views on that. Just to close for now, and I've got loads more, but we should stop. Healing. Is this a form of healing? Jesus says, is it lawful to do good or to do harm on the Sabbath, to save life or to kill? Dealing with, if you like, the law-based of not doing something on the Sabbath, ticking the boxes. And Jesus says, is it lawful to do good or to do harm? Is this harmful or is it good? And we could go on to talk about the possibility of using that for enhancement, not just for therapy and getting rid of diseases, but for enhancing human properties, make us more intelligent, make us more athletic, give us all sorts of things that we'd never dreamt of. But I'm going to stop there, and I think firstly we'll take some questions, and then we'll see how we go with Q&A, so whether you want to buzz to talk about things together. Ruth, I'll come back to you. Thank you. There's a hand at the back there already. Quickly up.
Thank you, Philip Young, first-year ordinand. Quick question to kick off with. Just have two questions. Quick question: is there a gene which predisposes one to homosexuality?
No. Right?
Thank you.
That's not to say there's not a genetic component, but
Right.
there is not a gene for homosexuality. There may be a combination of all sorts of genes that combine together to make one prone to that. Maybe. The jury is out for the science. There's a very good article written by Dennis Alexander, published in Science and Christian Belief, that just pulls together the evidence for that. It's not making any point from it, the evidence. So there clearly is some genetic component, but it's polygenic, not a disease for gene for homosexuality. And there is clearly an environmental factor that involves that as well.
Thank you. And the second question, you've done real justice to showing the complexity of the field, but I feel slightly ill-equipped to sort of navigate through it. A lot of wonderful questions that illustrate the complexity, but I would just be hugely grateful if somebody with a strong conviction in the truth of God's word and a love of life and all that sort of stuff to sort of try and navigate a path which may be one of many which could be a Christian response.
Which is why I've been very careful not to try and navigate that, because it is such a complicated area, and you'll find Christians of all persuasions giving a variety of different views. And I think we just need, the first stage is to actually understand the complexity, as you said, of that. And that you can find evangelical Christians who respect the Word of God, who take totally different views on it. You'll find those from a Catholic tradition taking totally different views. But I think the need to try to think through Christianly of what is a proper response to it, and to acknowledge that each of us then actually may come to a different conclusion. So I sympathize with what you're asking for, but I could give personal opinions. But I think I'd rather you had the questions and you were able to begin to weigh them up, knowing a bit of the science and a bit of the how we get where we do. There were loads of other questions. I saw a hand there, and then I think we'll come forward, and then over there.
Thank you, Keith. Earlier in your talk, you, I think, implied that you were unhappy about the influence of the organic lobby in agriculture. Could you unpack that a little bit, please?
There is... In terms of the organic lobby and their desire to minimize the use of pesticides and to grow things, if you like, naturally, I think that is totally laudable. The question as to whether organic food is necessarily any better for you than non-organic food. All food's organic, but it says he's speaking as a chemist. I think that is debatable as to whether it is actually healthier. The organic production, I have very great sympathy for the organic lobby in terms of, if you like, the output of whether it's better for your health. I think that is questionable.
To a complete ignoramus in this particular field, you've done a very good job, as has been previously stated, in sort of explaining the complexity of the whole subject. Do you see that the subject is going to become more and more complex in the future, and there's going to be more and more avenues in which Christians can find themselves going down, and it's going to become more complex to be able to understand?
Yes, I think it is, and I think the human genome modification that we dealt with at the end, at the moment it's a pipe dream, it's a fiction, it's not something that you're going to come across in any of your churches, probably, of somebody who wants to have a child that's genetically modified. But I think as we look 10, 15, 20 years down the line, this could become serious business. At the moment, it's forbidden. You may not do it by law. They can modify the genome of embryos in the laboratory under very precise controlled conditions, but they may not go beyond, I think it's the 14 days, and certainly couldn't be implanted. So at the moment, it's forbidden. That, of course, is that the law will be different in different countries. And there's always rogue nations and rogue scientists and rogue medics who will do anything for the glory of being the first one to do it. And once Pandora's box is opened, who knows what? So in that way, I think we need to think ahead of the possibilities. Now, what happens in 20 years down the line when you can choose your child as to whether they'll have athletic genes or musical genes or high IQ? I think it's science fiction at the moment, but the developments, certainly in the CRISPR-Cas technology, have been so rapid. The other genome modification techniques were inefficient and slow and very costly. CRISPR-Cas is efficient, it's precise, it's relatively cheap, it can be done in any molecular laboratory. And the implications of that could be profound. So I think we need to be influencing the debate that's going on in science and in medicine as to what should be allowable, so that we make sure that the boundaries legally are well set. I think, personally, using that to alleviate diseases, things like thalassemia, cystic fibrosis, Huntington's disease, I think those is a form of Christian healing. Right? And it's a form of healing for which Christians can affirm, that way around. But I think we do need then to have very clear boundaries as to what is permissible and what is not. And I think, I've just touched on it, the question then of what that says to those who are not genetically modified. And we've seen it, it was an outrageous statement from Richard Dawkins, and it's I think he's since retracted a little bit from it, of saying to somebody who was pregnant with a Down's child, was saying, we should have it aborted because it's going to be a cost to society. Right? And what does that say? If you've got a child, an embryo that is going to have cystic fibrosis, it's going to cost society if that person is born with cystic fibrosis. But that's still, what does that say about discrimination against that sort of person? Well, you weren't genetically modified. How awful of your parents not to do that for you, partly because it's you and partly because you're a drain on society's resources. And that's the implication that we could go down. You can see that sort of Brave New World.
There was a very powerful article, sad one, in the Colour Supplement. So there was a rather powerful and sad article in the Colour Supplement last weekend. A mother who had a child, 2-year-old with cystic fibrosis, would have aborted the baby, but wasn't given the results of a genetic test. And she was living with a huge amount of anger, a huge complex of emotions.
And yeah, terrible. And what that actually says to the child, that it's there in the press that says, well, I would have chosen to abort you. Oh. And we've opened the Pandora's box that would, only 30 years ago, would not have been a question. Now we know things. We know the likelihood of a child having that sort of a disease. What do we do with that knowledge? And sometimes the knowledge brings with it tremendous responsibility. What do we do? Thank you. There was a hand at the back there. And then over there.
Thank you, Sharon. Amusing family story. My daughter went, and I'm sure she won't mind me saying this, she went down the IVF treatment line. A donor was found. The profile arrived, and he was dark, blue eyes, and played a lot of rugby. But when it came to the religion box, there was no tick or no answer. So she phoned her mother up, and she said, "What am I to do, Mum?" And my wife, who's a nurse, said, "I don't think it really matters, darling. It'll be fine." Anyway, a few weeks later, another donor was found, and again, he was dark blue-eyed, very athletic, played a lot of rugby. And the religion box, he was an Anglican, so he must be all right. But it did bring the question of implantation of people of various faiths. I mean, if a Muslim lady had found a Jewish donor, or a lady from the Southern Baptist Convention had found a high Anglo-Catholic, I mean, interesting, aren't they? It doesn't matter, of course it doesn't.
It's fascinating, and that actually says a lot about the things that people ascribe to DNA that it just doesn't have. I think it was alluded to yesterday in one of the talks. There was a rather scurrilous book published in 2002 by Dean Hamer called The God Gene, where he said that's something to do with the VMAT2, which is to do with noradrenaline storage. It influenced people's spirituality. I have to say that has never been published in the peer-reviewed scientific literature. If it has an influence at all, it is very, very, very small. And it's self-evident, I think, that religion is not in the genes. Now, there are those, you can see some families of fifth-generation pastors, and you see them advertised, particularly American websites. We're a fifth-generation pastor reading us today. And I think, what I don't care about is what's gone before. What's their beliefs now? And you can see very devout Christian families whose children turn away from the faith. And similarly, you find atheist parents who have Christian children. Francis Collins, I think, once said that he became a Christian from being an atheist in his mid-20s, and there's no evidence that his genome changed. There's the influence of genetics on religious belief is minimal, to say the least. Environment, all sorts of other things play a part. Can I take? Actually, I did promise one over there, and then I'll come back.
Yeah, I think one important point that's come from all this is that information is dangerous. And don't you think that the main thing that Christians and people of faith can bring to this discussion is just bringing the issues to the fore and presenting the moral conflicts that are going on? Because, as you've rightly said, rules are going to clash. And there may be lots and lots of different rules clash. Where do you go from there?
I think that's really important. It's actually people being informed about the science, about the ethical principles is actually very, very important. And using, which is why I put the words up about now the Frankenfoods, actually some of the pejorative terms that we use out there are just not helpful in terms of getting to grips with people who don't understand the science and don't understand, if you like, the ethical principles, let alone the Christian moral precepts, and getting information out. And you as church leaders, I think the responsibility is on you to better understand what are the issues at stake, rather than just take, if you like, what I might call the Daily Mail knee-jerk of no, it must be wrong, or it must be right. Getting to grips with the issues is a tremendous responsibility. Now we have the knowledge. Thank you. There was a hand over here.
Can you say something about, I think that my schoolgirl science suggested that our genes were fixed at birth. And I now know that that's not correct. There are some things that are given to us, but there's lots that isn't. There's lots that never is activated, and there are some things that are activated. And we don't understand all the reasons why some genes are triggered and some aren't. But there are also viruses that come in that change us, which the fact that that there are these unknowns and influences which mean that our genes aren't entirely set,
Right.
suggests it shifts the debate away from,
Thank you.
it shifts the debate.
Yeah, thank you. Our genes are set, but our genes are modified. And so your DNA sequence is set at birth and nothing will change that. Well, mutations can, but there are tags that are put on your DNA sequence which are influenced by the things that you eat, by the social environment that you're in, by the stresses that you go through. And those tags are what we call epigenetics, and those affect which genes are expressed, so expressed is a sort of coded word, are used in different times. And that actually relates a little bit to what Alistair Coles was saying yesterday about the ways in which our habits self-reinforce. That actually comes back sometimes at the level of our genomes as well, about the things that we do, the things that we eat, the people that we meet with, do in some way affect those tags that are put on our genes that alter which ones are expressed. And you can see that in some ways, in not a trivial way, in terms of the things that we eat. There's been quite a number of studies on what are called developmental origins of health and disease, and that has originated, some of that, out of Southampton University, where I'm privileged to be part of my time, is that the status of the nutritional status of the mother will affect the child as they grow. And that's not to do just with their genomes, but the way in which epigenetics. And what happens, let me just take you through what is, if the child is starved in utero, the tags are put on the DNA that says, effectively, make enzymes, make proteins that grab whatever you can and keep it, right? Because nutrients are precious. Now, if you take that starved child and then put that in a nutrition-rich environment, the tags still say, keep whatever you can and grab hold of it, because you never know when you might get to starvation again. And those are the children that are more likely to suffer from heart disease later on. So you see the genetics have stayed the same, but the tags that are put on are different. And how long those tags stay on for is an open debate. There are those who say actually that these sort of epigenetic phenomena can be inherited for second and third generations. Doubtless that they do fade with generations. They're not fixed like genomes. But I think it's very important to emphasize that our genes, again, are not us. And we're influenced by the people we speak to. I think, on a different talk, I use an African quotation. And I think it's that a person becomes a person through persons. It's the interactions that we have with other people. It's no man is an island. It's the interactions we have, the relationships we have that make us people. That's restricted sometimes by our genetic makeup, but it's not dictated by it. We could go on for a long time. Thank you for that question. We'll take one question over there, and then I want to give you just a little bit of time to discuss together.
Is a concept like human flourishing too broad a concept to be useful in helping Christians to make decisions about these sorts of issues? And for instance, if one tries to apply that to an individual, would it give rise to very different consequences from trying to apply such a concept to the species as a whole? Yeah. And I wondered what you felt about trying to give some way of helping Christians to make such decisions, and whether something like a concept like human flourishing is at all useful.
I thank you. I think that's really helpful, because I question that. Flourishing for who? Flourishing for the individual or flourishing for the society? Sam Harris, one of the new atheists, wrote the book The Moral Landscape, where he tried to make a scientific case for human flourishing, giving moral principles. And I think we could talk again about that at length. I think he failed within that book. And I think as Christians we need to think very carefully too about whether it is the flourishing of society or the flourishing of the individual. And you can pick up Bible passages that point in opposite directions in that. Is God's concern for mankind as a whole or for the individual? I think in the Western world we've probably overemphasized the individual, the personal. In other parts of the world it's society and mankind as a whole that's emphasized. And I think we need to hold those in tension, and that's very helpful, what you said, is the phrase human flourishing by itself. I'm not sure how helpful that is. Thank you. Can I stop questions there and suggest that we've got maybe 10 minutes, and you sort of break into groups of people around you and actually ask the question, what's the difference between enhancement and therapy? What would you be prepared to edit out, and what would where should we not go? Very loose question. You can talk about anything else if you want to. What you had for breakfast, if you really want. But just ask, where would you draw the line? What's the difference between therapy and enhancement? What's a disease that's worth healing? Over to you.